Hi, everybody. Proteins Part 3. First one we did was about the amino acid structure on its own. Our second video was about how to covalently bond those together by dehydration synthesis. And if you don't remember that, that's actually the process that happens by ribosomes in your cells. And what we're going to cover today is how you actually take a linear chain of amino acids, as depicted on the board here, and form complex shapes like this ridiculous thing or a pair of scissors, but that's for later. The first thing I want to remind you of is that the linear arrangement of amino acids is called a polypeptide. And I should remind you that it does have direction in that the first amino acid that leaves a ribosome when a chain like this is being produced is the one over here with an exposed amine end. And so, as I mentioned last time, that's why this is called the N terminus, because an amine has a nitrogen in it. So this is the first end that comes out of a ribosome, and this is genetically important. So the N term or N terminus. And the reason this is important genetically is when you're reading a gene sequence in order, the first three nucleotides in that gene sequence, the AUG, for example, indicate the first amino acid that's included in the polypeptide chain. So the first three nucleotides would indicate this amino acid. The next three nucleotides would indicate this amino acid, et cetera, et cetera. So that's the connection between the genetics and then the actual protein structure back here. Okay, so what we have is an N terminus at one end, and at the other end, we have a carboxyl group. That's the tail end of the polypeptide, and we call that the C terminus because it has a C in there. And I can't reach because of the cord, so I'm not going to actually label that C terminus. All right, what I've not labeled in this whole thing are the R groups. I've left those out. That's these blank bonds with no R on them. We'll get to that in a moment. All right, what I do want you to notice is that once we've strung a bunch of amino acids together, this linear structure here is what you can call the backbone of the polypeptide, right? This is just the linear string that holds it all together. And you'll also note that we've got amine and what are called carbonyl groups alternating around those central carbons. So amine, carbon, carbonyl, amine, carbon, carbonyl, amine, carbon, carbonyl, amine, carbon, carbonyl. What's also really important to notice about this linear primary structure is the amines and the carbonyls are polar. They are quite polar groups. The amines, which are an NH, which I didn't picture there, and a CO, these are polar groups. The H off the N is a little bit positive, and the O off of the C is going to be a little bit negative compared to the N and C themselves. So these have the possibility of hydrogen bonding. We'll get to that in a second. So there are going to be four levels of protein structure that we're going to be discussing in this class. The first level we just talked about, just this linear arrangement. When you make those covalent or peptide bonds, and peptide bond is just a specific name for a type of covalent bond between amino acids. It's just annoying vocab. When we string the amino acids together, this linear arrangement is called the primary structure. It is literally just amino acid 1, amino acid 2, amino acid 3 in a linear fashion. So if you see something written out as methionine, alanine, valine, glycine, tryptophan, tyrosine, those are the names of amino acids. That is the listing of a protein's primary structure. And so you just have, in model form, just something like this, just a tube. Flexible, floppy, no real structure to it. At one end, an N terminus with a blue cap. At the other end, I can add a red cap to represent the C terminus. So how do you form structures? Well, because the backbone itself, the amines and the carbonyls, are actually polar, and because the slightly positive H off of an amine and the slightly negative O from a carbonyl might actually form hydrogen bonds if they come into the right orientation, what you might find is that this will actually start to fold into temporary, floppy, what are called secondary structures. And so I'll demonstrate the two most common ones that we're going to discuss in this class. The first is known as an alpha helix, and it's that. And what you're getting are these vertical hydrogen bonds between an amine of a backbone part here and a carbonyl here. So a hydrogen off of a nitrogen and an oxygen off of a C. And so those are going to form a weak but significant bond, especially if you get 30 or 40 of them holding this structure together. So this is an alpha helix. The other type of structure that you can get just by H bonding the backbone is where the backbone actually folds back and forth. Or even more complex than that, you can get these wider folds, but eventually it folds back upon itself in parallel, and this is called a beta sheet, or a beta pleated sheet is the longer version of that terminology. What you need to know about here, overall, this is all called secondary structures. An alpha helix is a secondary structure. A beta sheet is a secondary structure. You can think of this as being kind of like a beam and a wall, sort of a more linear shape and a more flat shape. And by using 2x4s and sheetrock, you can build a whole lot of stuff. For example, a house, notwithstanding the foundation. This is the basis for creating a more complex structure. The bonds that are holding this together are the H bonds, the hydrogen bonds between groups of the backbone itself. We still have not talked about R groups. Okay, so this is going to be a bit of a long video, so bear with me. That's the second level of structure. Primary structure, just the covalent peptide bonds holding the amino acids together in a linear fashion. Secondary structures are all of the H-bonded backbone structures, the alpha helices and the beta pleated sheets. Okay. Third level of structure. Well, what about the R groups? So off of these structures, off of the backbone, there are R groups popping off. And so I could add some on, and I'm not actually choosing specific ones out of necessity right now. But I'm just popping on R groups. And each of these R groups has a particular structure to it and particular properties. And depending on the shapes and the properties of these R groups, they may or may not be able to bind with one another. And that's the key here. Because if they are able to interact with one another, these shapes might start folding back upon themselves, forming more complex structures. So tertiary structures are formed when R groups form bonds with each other, basically pulling alpha helices and beta sheets closer together and stabilizing them into a more complex arrangement, like so, or like so, or like so. So what kind of bonds are we going to form? Well, it depends on the R groups. If you have two polar groups, R groups, you're probably going to be able to form a hydrogen bond. So for example, this right here is a polar R group. This represents a hydroxyl group. And if we had another group over here, this one over here is polar. It's actually acidic. But these two would be able to form a hydrogen bond. Okay. Now, what if you had a positive R group, such as this one, a negative R group, such as this one, and a positively charged R group, such as this one? Well, if you had a positive and negative, full charge, you might be able to actually form an ionic bond. What if both of those groups was nonpolar? Well, that's what's shown here. This is all carbons and hydrogens. This represents something that's all carbons and hydrogens here. In that case, they can't form charge-based interactions. And so what's going to end up happening is they're going to get shoved away from the water and all of the other polar structures. And this one's a little bit harder to understand. But if they can't interact with anything else, if these non-polar R groups literally cannot interact with the water or any of the polar groups, they will be rejected by the water and all of the polar R groups and the backbone. They will be rejected by all that, and they will be forced to go as far away from everything else as they can. And the easiest way for them to do that is to go to the very center of the protein, hidden away from the water, hidden away from all the polar groups. So typically in a lot of proteins, you're going to have in their centers, in the sort of core, a cluster of hydrophobic or non-polar R groups in the center. Not that you can see that so well here. So to recap, tertiary level structures are the interaction of secondary structures via their R groups. So an alpha helix over here might interact with an alpha helix over here. An alpha helix over here might interact with a beta sheet here. A beta sheet here might interact with a beta sheet here by means of hydrogen bonds, ionic bonds, or even hydrophobic interactions, van der Waals forces, if you want to use those terms, between nonpolar groups. So these are going to be R group interactions bringing secondary structures closer to one another. Okay, so this ugliness is completely nonsense now, but that gives you an overview. Last thing that you need to know, there's a fourth level of structure, and I'll just show you an example from reality. And here's one. Take the R group off of it. This doesn't look like a protein because it's not. But what I'm showing you is that this functional structure actually has multiple pieces. This actually has 3 pieces, 3.5. You might look like 2. You've got this pair of scissors is like one scissor and another scissor. There's actually the blade and then the anvil. And then there's actually a rivet holding them together, which you could consider 1 or 2 pieces, however you want to describe it. It's really 2. But there are multiple parts all put together to form a functional moving structure. Okay, a functional structure. Most proteins are like this too, right? Literally, most of the proteins in our bodies have multiple pieces. Like a tong wouldn't work, but a pair of tongs held together by a rivet actually has a function. And the same thing is true of proteins. So oftentimes, for a protein to do its job, you need to have both parts of it, and then it can act as a clamp or a stapler or a motor or something functional like that. And so that means that this polypeptide chain, which has got all levels of structure, primary, secondary, and tertiary, that we've discussed so far, might actually have to bond to another polypeptide. Oftentimes they're identical, not always though. And the interactions here are going to be the same as the tertiary ones. It's going to be R groups. So what's going to end up happening, typically, is this will also have R groups exposed that haven't formed bonds. And those R groups will interact with these R groups on this that are exposed, and these 2 polypeptides will bind together using H bonds, ionic bonds, etc., to form a functional protein. Okay, let me recap because this is a long, long video. Primary structure is just covalently bonded amino acids, covalent peptide bonds between amino acids forming a linear, floppy structure. No structure really beyond the linear nature. Secondary structures are those alpha helices and beta sheets formed by hydrogen bonding between backbone groups, between the oxygen of a carbonyl and the hydrogen off of an amine group here. Certain number of turns down. So those are alpha helices and beta sheets. When you start taking the R groups into account, and when secondary structures start to bend and fold towards each other, that's tertiary structure. And the most common types of bonds are going to be H bonds between polar R groups, ionic bonds between positively and negatively charged R groups, or also basic or acidic R groups, if you want to think of it that way. A third type of interaction would be hydrophobic or van der Waals interactions, which is basically the inability to form anything else. And that's going to be formed between nonpolar groups, often in the core of a protein. The one that I have not discussed is called disulfide bridge. We'll consider that another time. And then tertiary structures are the same kinds of bonds as you'd find in quaternary level structure, same kind of bonds as you'd find in tertiary structures, only now we're forming them between multiple polypeptide chains to form your final functional protein. That's the end of this overly long video. Watch at your leisure, rewind as necessary. I hope that helps. Bye.